and I'd like to introduce our panel moderator. That's Dr. Ryan Carnegie. And Ryan is a shellfish pathologist and parasitologist and leader of the Shellfish Pathology Laboratory at the Virginia Institute of Marine Science. He conducts wide-ranging research, advisory service, and student training in the evolution and ecology of marine diseases, parasite phylogenetics, and phylogeography, and practical shellfish health management in aquaculture and fisheries. He has been studying oyster diseases in the Chesapeake Bay region since 2003. And so with that, I'll turn it over to you, Ryan.
Okay, thank you, Raleigh, and thanks to all in the audience for plugging in, as Emily says, to this second part of this morning's session. So by way of introduction, this is the panel on oyster fisheries, restoration and aquaculture in Chesapeake Bay, critical research and management issues. So just to provide a bit of introduction, oysters, of course, are an essential part of the fabric of the Bay region by virtue of the roles that they play in the environment and with respect to their importance to coastal communities going back over millennia. We obviously care a lot about them. We're heartened that they are doing better after their depletion due to overharvesting and habitat destruction and disease over the last 60 years. And there's increasing confidence, I think, that in many ways, with regard to oyster resources, we're on the right track toward seeing them improve. If we can maintain investment in restoration as a priority, if we can produce enough seed, if salinities remain high enough to sustain good recruitment, but not so high that we lose many of them to disease, will be okay. Now, we could have had a panel on the progress we're making with oysters, and we are making progress, but I think it's more interesting and really important to consider where the flaws are in our foundations with regard to managing these resources. What don't we know that we need to know to manage oyster resources more effectively? What are the questions we need answered to better understand how these populations work, how they fit into our Chesapeake Bay ecosystems, how oyster populations in aquaculture and the wild are interacting with ecosystems? Where are we challenged and where are we limited in our ability to manage oyster resources by this knowledge that we don't have? Some of the dialogue we've had around this conference was about engaging early career scientists in oyster science and bay science generally and highlighting opportunities. In this regard, it's far more instructive not to simply express that we have all this under control and that anyone new in the field should seek fertile ground elsewhere for their scientific exploration. Rather, it's better to send the message that there are urgent questions still needing attention and awaiting or requiring new expertise and new perspectives and inspiration that new scientists will bring to the system. So this was the spirit in which we embarked on this panel. What I'd like to do is start with some brief self-introductions from the 6 panelists who are on the panel. They include researchers representing diverse areas of oyster science, as well as professionals working in the Bay region where the rubber meets the road at the interface of science with activities like fisheries resource management, aquaculture, and restoration. So for the self-introductions, we'll start alphabetically. We'll proceed alphabetically, and we'll start with Allison Colden from the Chesapeake Bay Foundation. Allison, could you go ahead?
Yes, thanks, Ryan, and good morning, everyone. My name is Allison Colden, and I am the Maryland Fisheries Scientist at the Chesapeake Bay Foundation, where I work primarily on oyster fisheries policy, but also assist in our oyster restoration programs. Thanks.
Okay, thank you, Allison. Jeff Cornwell is next.
Hi, I'm Jeff Cornwell from the Horn Point Laboratory at part of the University of Maryland Center for Environmental Science. I've been working on oysters for a couple of decades on water quality issues, originally with Roger Newell, but more recently with Mike Owens, Lisa Kellogg at VIMS, and Allison Colden. I'm currently the Vice Chair of the Maryland Aquaculture Coordinating Council and Chair of the Oyster BMP panel.
Okay. Thank you, Jeff. Karen Hudson.
Hi, everybody, and thanks, Ryan, for the opportunity to be on the panel. I'm Karen Hudson. I work at VIMS. I'm the Shellfish Aquaculture Extension Specialist. Been at VIMS for about 20 years, in the last 10 in extension, and so the main my main task is really to be that bridge between, you know, all the great science and research that's going on with shellfish culture and the folks that farm shellfish, oysters, and clams on private ground. And so that's my main focus, is on the private aquaculture community. Thanks.
Okay, thank you, Karen. Judy, I believe, is calling in.
Yes, thank you. Good morning, everybody, and glad to be here. I'm Judy, the Shellfish Division Director at the Maryland Department of Natural Resources. And we've been involved with fishery and sanctuary work for, well, decades, actually. I've been there many years. So we have some, how'd you say it, where the rubber meets the road experience. And we also work, of course, with the university and other researchers and the environmental groups. Thank you.
Okay, thank you, Chris. Juliette Smith is joining us.
Hi, everyone. Ryan, you can hear me okay?
We can hear you. We can't
Excellent.
see you presently, but yeah.
Oh, how's that?
That's
there we
better.
go. Sorry,
Yeah.
everyone. Good morning, everyone. I'm Juliette Smith. I'm an associate professor here at VIMS. And I would say I sit somewhere between a habicologist and a toxin chemist. My research with oysters has really focused in the last 6 years. Prior to that, I involved oysters, but I really thought of them as kind of the possible vector of toxins up the food web to higher trophic levels or humans. And I would say since joining VIMS in Chesapeake Bay, I have a much greater appreciation for oysters as a key organism in the system. And so now my research focuses not only on oysters as a possible vector of some of these contaminants, but also what about the environment could impact the oysters, and how can the oysters then go back and impact the environment? So I have a brand new appreciation for oysters, and thanks for letting me join the panel.
Thank you, Juliette. And finally, Mike Wilberg.
Thank you, Ryan, and thank you for inviting me to be on the panel. I'm Mike Wilberg. I'm a professor of quantitative fisheries science at Chesapeake Biological Laboratory of the University of Maryland Center for Environmental Science. My main focus of research on oysters is on looking at or trying to describe their population dynamics, how they respond to fishing and other sources, and how potential other policies for managing or trying to restore them will play out into the future. And so I've been involved in the Maryland Oyster Stock Assessment, the Oyster Futures activity that involved a group of stakeholders to try to explore potential policy options for restoring oysters in Maryland, as well as a variety of other things for about the last almost 15 years now. So.
Okay, thanks, Mike, and thanks again to all of you. I have submitted a number of questions to the group in advance for their consideration. And we do want to have something of a free flow to the discussion here. I would remind the audience that we still have the question and answer function available. If you have questions you'd like to pose to the panel, please do post them to the Q&A box, and we'll see if we can get to them. But to start the dialogue off, I think we'd like to start with the first question that I posed. What are the critical research issues or gaps that we need to address? And let's start with Mike Wilberg on this one.
Thanks. So these are the ones I came up with for today. They switched around a lot depending on which question I've been asked recently. But I think one of the really big open questions that we still have is to get a finer understanding of how oysters interact and affect their bottom habitat, and then how we can use those interactions to best promote their restoration, as well as to manage fisheries for oysters. And so, for example, some of the things that I think we don't have a very good handle on now are the first part is just even how much oyster habitat is out there. And I'm thinking particularly here, bottom substrate. How much shell do we have in the system? How is that shell changing over time? What factors are causing it to decline? What things are allowing oysters to cause to increase? What are optimal restoration strategies? And so we've been learning a lot in recent years, but I don't think we still know exactly what would be the best course of action to pursue for oysters in any given area. what does sustainable harvest look like for oysters? I think this is still in its infancy. In terms of most of fisheries science, we talk about sustainable harvest of species, but we don't think about habitat effects of that harvest, whereas oysters, that has to be brought to the forefront, and so it's another thing that's pretty much linked. So I think those are the top of my list for today, at least.
Okay. Thanks, Mike. I think we'll just continue through the group with this first question, and move on to Juliette next.
All right, thanks, Ryan. All right, so major research gaps. So looking through my lens of environment and harmful algal blooms and their toxins, I feel like I'm still synthesizing all the literature on this topic in respect to oysters in Chesapeake Bay. But for me, not only when looking at oysters, but also in the larger context, I feel like for me, the biggest gap is really that we need to look towards multi-stressors. So I feel like we're all understanding and realizing that we need to move away from kind of these single factor experiments in the lab or in the field, where we're just looking at things like just temperature or just pH or just salinity or just Hab cells or their toxins and their effect on oysters. I really feel like now we need to be looking at combinations of those, and those experiments are absolutely more difficult to run, but I do think that they're worth running if run with the appropriate controls. And that's because I really don't think any of these, and as I'm sure you don't, think any of these factors are changing in isolation. And I feel like if we really want to understand how these different factors are affecting our oysters now and then into the future, we have to start doing these complex multi-stressors. So I guess I would propose things that are kind of designing experiments or evaluating the data with respect to multiple algal toxins at a time, because those aren't occurring in isolation. You can have more than one harmful algal bloom at a time, especially in Chesapeake Bay. Things like changing pH with temperature and with salinity and with different algal toxins present, or different PAHs or other contaminants present. And then trying to think towards now, how would that change toward the future? So for me, that's a major research gap that I would hope we all continue to explore.
Okay. Well, thanks, Juliette. We'll definitely circle back to that point. I've got some notes that I'll return to, but we'll work through the rest first and circle back around. Jeff Cornwell.
Yeah, so I mostly work with transformations of nutrients associated with bivalves, and I'd identify two areas that are ripe for a better understanding. One of which is when we're looking at bottom reef environments and oysters on the bottom. When we make measurements of denitrification, there's a high degree of variability, and it's only partly accounted for by changes in oyster biomass. We also know a lot about denitrification in the sense that we know it tends to move to the clumps of oysters when we excise them from the sediments, but we don't have a complete grasp of the factors that control the amount and efficiency of denitrification on the bay bottom. We have overall rates. We can tell you a lot about what happens in a square meter, but we can't exactly tell you how this all happens. There's still a major fundamental scientific research gap. The second thing I would point out is that oysters and what they do in terms of biogeochemistry, it really comes down to the fate of the biodeposits, the feces and pseudofeces. We don't really know much about their production, transport and resuspension, and ultimate fate in terms of their decomposition. And so to really have an understanding, I think this becomes especially important as we're looking at the effects with aquaculture. We need to know where these particles, how these particles are assembled, and what their fate is. Do they get denitrified down the line? Because the footprint of an aquaculture site may not be reflective of the overall net nutrient balance. Thanks.
Thank you, Jeff. Let's move on to Karen Hudson. So research issues, research gaps, or challenges at the interface with management, whatever you're thinking about, Karen.
Yeah, thanks. Well, through my aquaculture specific lens, I mean, I would definitely say, you know, water quality for the hatcheries, for hatchery production is crucial. We've done a lot of work, a lot of good work. We've come a long way. And to Juliette's point, you know, we really started out focusing on single issues, whether it be carbonate chemistry or harmful algal blooms. We've really worked to get to a co-stressor research, but we're just still not there where we can say to hatcheries, hey, you know, this is your problem and here's how you manage around it. You know, they can't manage something we don't understand. And so I would just say that, you know, this is something we're working on for sure, but it's still, you know, something that's a big gap, because, you know, for aquaculture to expand to be sustainable and to, you know, potentially expand, we really need the hatchery production. And certainly I would think in Maryland, you know, for understanding production problems is important in restoration as well. So I would say that one. You know, another one on that same track, the carbonate chemistry, the OA or coastal eutrophication and harmful algal bloom toxins. We've spent some time looking in the, you know, inside production facilities, but I mean, to me, and maybe it just may be naive where I sit, but, you know, I'd like to know what is the impact on the, you know, on the wild oyster population. What's the impact? Are we seeing impact, you know, on the farms? You know, I think we certainly know there's issues inside a production facility, but what's going out on, what's happening out in the environment? I think that's, to me, would be something that we need to get a handle on. And I think just one more is this SAV and aquaculture, determining the impact of oyster aquaculture on SAV for a management. You know, that kind of comes up in another question, but, you know, to address management issues, we really need to understand that. We need to know, can these things coexist? Can we manage them in different ways? And the only way we're going to be able to do that is with data. And again, we're working on it, but I'd love to see more on that. Thanks.
Thank you, Karen. Allison?
Thanks. I think a lot of great points have already been made, so I just want to provide a little bit of perspective too. I think Mike touched on some of these as well as Karen. You know, I think one of the things that's unique about Chesapeake Bay oyster restoration and our approach is the scale at which these projects are happening is relatively unprecedented and compared with some of the other estuaries in the U.S. and even overseas. And I think that is a benefit, because obviously I think that the data is starting to show that is something that is functioning for the most part, but it also means that we're sort of out on the cutting edge with few other examples worldwide to look to in terms of understanding the larger ecosystem context. So one of the research questions that I have is sort of the question of scale, and how big is big enough when it comes to oyster restoration, and what sorts of scale would we need to be working at to start to see an ecosystem wide or Chesapeake Bay wide you know, influences either on the ecosystem or on the oyster population, because I think sitting in as a member of the Maryland Oyster Advisory Commission and other management contexts, it's easy to qualitatively throw out the goal that we want more oysters and we want self-sustaining oyster populations, but it's hard to know, hard to translate that from sort of a qualitative and societal goal to how you get there from a restoration and management context. And I think that comes to another research question. You know, Mike mentioned the Maryland Oyster Stock Assessment, which is a huge leap forward in terms of scientific understanding of how the population is working in Maryland. And the question that I have as well is, how does that translate into management? And I think that gets back to the issues of trying to understand how the fishery impacts not only the populations, but as Mike said, their habitat, and what the feedback loops are there in terms of trying to not only manage the numbers of oysters, but also maintain some sort of semblance of habitat to allow for recruitment into the future that supports the fishery. Understanding the larger landscape context, and again, this is something Karen touched on, understanding where oyster reefs fit in the larger ecosystem context. And one thing I'm really interested in is not just from a management context, the interactions of SAV and oysters, but also trying to understand how we could possibly leverage the feedbacks between these types of ecosystems. You know, if it turns out that the relationships between SAV and oysters are positive feedbacks and are facilitating one another, is there a way that we could leverage those to get a better bang for our buck from the restoration context ecosystem wide? And lastly, you know, I think another thing that's important, and this is more from a management context, but it's informed by, hopefully informed by science, is understanding what the end point of oyster restoration should look like. Again, we all have these sort of policy and societal goals for what we hope oyster populations can do and support in terms of their ecosystem and their fisheries support, but taking that and translating it in a scientific way would be helpful in guiding management and restoration in the future. And I think that's another big data gap that we have.
Okay, thanks, Allison. How about Chris, Judy?
Yeah, well, thank you. So, so many good ideas have come forth. That gives me a short list because a lot of it's already been said. One thing I think we need to look to is a better knowledge of our bay bottom. We do bay bottom surveys when we undertake a tributary like Minokan River or Little Chop Tank, et cetera, but we don't have a bay-wide comprehensive set of maps that show sand and mud and a mixture of sand and mud, shell, oyster bar, et cetera. So redoing the bay bottom survey would be very helpful in plugging that gap. The last one was done in the early 1980s. That's 40 years ago. Another topic that's, I think, helpful, but maybe hard to achieve would be, is there any way to learn more about larval tracking and what the sanctuaries might be providing as a far field effect? You know, there was a lot of hope, there still is a lot of hope that sanctuaries will produce larvae and spat outside their borders. So any kind of research to that would be helpful, but I'm sure that's very hard to track down. My third comment would be, and final, you know, habitat, of course, is crucial for oysters. They need habitat, and they also make habitat. And we don't have much shell, as everybody knows. It's hard to get. And when we do get it, it's in short supply. So two ideas that maybe are data gaps and also research needs. One would be developing an artificial culture. Would it be possible with the right formulation, something that could be used in the bay safely, environmentally, and with a factory, actually make something that looks just like an oyster shell? We could use molds that are shaped just like bay shells. So would it be possible to manufacture large volumes of culture? Now, of course, society would want to speak in. Do we want to put this artificial material in the bay? But I would put forth it's better than perhaps recycled concrete, cinder blocks, and ground up toilets and such. So the challenge would be, could we develop and actually manufacture large volumes of artificial shells? But another idea that's more natural would be, would it be possible, and this could be a research project, would it be possible to overplant spat on a site? So as a quick example, we plant 3 to 4 million spat per acre to do a restoration project in a sanctuary. But first we put down about a foot-thick layer of material, shells or other material, and that's costly. And there's a lot of issues with the material we might use, rocks and such. So would it be possible to maybe put down a thinner layer of material, save that money, channel that money over to the spat production, and instead of planting just 4 million spat per acre, perhaps 8 or 10? And the reason I say this is there's only one entity that I know of in the world that actually creates oyster shell. It's not the government, it's the oyster itself. So if we overplant spat, would it actually be beneficial shifting the money to that, shift money to overplanting spat and getting them to create the shell? And here's an observation that made me think about this. When you grow oysters at a pier in a cage, the cage is hanging from the pier. you have a certain density of spat. Homeowners do this, for education and outreach and such. And it's quite commonly seen where the oyster spat cement themselves to one another, and you have this almost John Smith type solid reef structure in this cage. So my point would be, could that be duplicated on the bottom, where oysters being overplanted at high densities actually create their own hard integrated substrate? I think that's worth looking into.
Okay, Chris, thanks. So Chris mentioned the issue of sanctuaries and the how are they performing, larval, tracking, possibilities and so on. And this is a big question. How do we understand the function of sanctuaries, whether they're working as we would hope them to be, whether they're worthwhile, investments in conservation and financial allocations and so on. Does anybody have any thoughts on how to get at the issue of sanctuary function? Are they doing what we hope them to be doing?
I can give a little bit of an update on how sanctuaries are performing in Maryland, and the short answer is it's complicated. And so I tend to think of sanctuaries in kind of three groups. We have the ones where there's major restoration efforts that are going on, and those ones tend to be quite successful in terms of increasing oyster abundance. So that so far would be Harris Creek, the Little Chop Tank, and Tread Avon. For the other sanctuaries that haven't had major restoration efforts in Maryland, we have a much wider disparity of performance. So some of the sanctuaries in the southern portion of Maryland, so the Minokan River, the Nanticoke River, the upper St. Mary's River, those ones are all actually doing quite well. And so we're seeing positive population growth of oysters in those areas. A lot of the other sanctuaries that are in lower salinity environments haven't really responded the way we would have hoped. And so it points to the idea that fishing hasn't been, at least fishing in that area currently probably isn't the driving factor for what's governing their performance. And so in many of the areas in the northern part of the bay, we haven't seen recovery of oysters, and we're still seeing decline of oysters in many of those areas. And so with regard to larval transport, we've modeled it, but we haven't been able to verify whether our models are correct. If our models are correct, there should be a substantial subsidy coming out of the areas that we've restored, but we still haven't quite gotten to where we're able to actually identify which oysters come from a sanctuary versus which ones came from outside the sanctuary in order to be able to really test that well.
How would we do that, Mike? Are you thinking, would that be a genetic exercise, or?
I think a genetics would probably be the only feasible way at this point. Elizabeth North's group with Jason Spires have been looking into some sort of dyes that could be used with larval oysters that could also potentially be used, but that one is a little bit, I think is a little bit trickier, but there's some potential there as well.
Mm-hmm. Okay. So Karen Hudson mentioned water quality being a key issue with regard to, well, really so much that's going on in the Bay, and certainly in aquaculture production. Why do we have such a hard time, for example, producing oyster seed as we get into the middle part of the summertime? I don't think we have a good sense of what poor water quality is. Is that something that we could figure out? Is there a way we can get at what is poor water quality? What is it about water quality that's limiting to oysters? Do we have any sense of how we might do that? I don't know if Juliette or Jeff perhaps might have a thought in that. How do we get at this issue of water quality, and what is limiting?
I don't work directly with hatcheries, but I've talked with our hatchery people a lot and been part of a couple of proposals. And the difficulties that hatcheries have aren't necessarily falling in. And this was mentioned before. The obvious things about pH, contaminants, other things of that kind, just don't show up as a limit to producing spat. So there's something else, and we just haven't gotten to it yet.
So is there one something else, or as Juliette suggested, is it multiple stressors interacting, and we just don't have a firm grasp of the interactions yet?
No idea.
I would say for me, it was very enlightening to get within a hatchery. And for me, I think that was my limitation until I actually was allowed and welcomed into hatcheries, that once we were able to do kind of more of a consistent monitoring of the within hatchery water, it was hard to really understand how all those different things might be crossing. But once we have now moved research to that level, and there's challenges with that in itself, but it's been really eye-opening. And now that I can see how pH is changing relative to temperature as we move through the summer, how algal toxins are making it into the hatchery, which I had always assumed that by putting this, series of water treatment in place, so that the water being fed into the hatchery, was going through different levels of filtration or diatomaceous earth or carbon filtration. I did not expect to see something like algal toxins making it into the hatchery water. And not as a concern for human health, but as a concern for animal health, right? For the oysters, growth and survival, or even, relating to algal crashes. So for me, getting within, being invited to within the hatchery and having this like regular monitoring has been really key. So I think before that, it was a bit more of a like, something went wrong and, we're coming in to kind of try to help, but the event has already passed. So being brought in earlier and kind of almost in a preventative or research monitoring sort of way has been really helpful for me from understanding water quality within a hatchery.
Okay. Allison, you mentioned it among your points, this, how big is big enough with regard to restoration, the scale of restoration, which I, suppose, speaks to where the resources are going and how they're distributed, resources not being unlimited for restoration in the region. How do we determine how big is big enough, or how small is too small as to be not worth an investment? How do we figure that out?
Yeah, that's a great point, Ryan. I think, where we have been in the oyster restoration community over the past 30 years, I think where there's been a shift is from a lot of smaller projects that were sort of scattered more throughout the bay with the Johnny Appleseed type of approach, where if we do a little bit of restoration everywhere, has sort of shifted with the Bay Agreement goal of restoring 10 tributaries by 2025, has really shifted the goal into sort of a larger context in terms of scaling restoration. How you determine how large is large enough is a question that I think still remains. And the other question that I have too is we're starting to see, particularly in Maryland, shifts in productivity, where areas that were once extremely productive oyster grounds haven't been for a long time. I think we also need to have a larger ecosystem viewpoint of what is the current suitability of various areas now and under different scenarios of climate change moving forward, so that we can prioritize and target those limited resources, like you said. I think the other thing that you touched on, which is super important moving forward, is being able to quantify the ROI, the return on investment, that's based on the ecosystem services in restoration. Especially where restoration is happening within a sanctuary, we don't have the opportunity to quantify the value of oyster population increases in the traditional context of increased harvest, which everyone is familiar with. And as many on this webinar probably know, it's quantifying those ecosystem services and taking it that one step, that one connection higher, which is where things get so much more difficult. The results get so much more variable, and it can become very difficult to have a clear demonstration between different localities and geographies of that return on investment. But I really think that that work continuing to move forward is going to be super important, just because, as you mentioned, this is not a cheap thing to do. Oyster restoration is not inexpensive. The momentum that we'll need just to complete the 10 tributary goal is going to require a lot of resources, and being able to understand and communicate the return on those investments to the public and to policymakers is going to be important to making sure that that momentum can continue. And the larger question of how big is big enough to sort of see a system-wide change sort of puts an endpoint on it. It can be very difficult from a just speaking now as my role as an advocate, it can be very difficult to convince someone that resources are needed if you can't tell them when it stops. When does it ever end? And you know, that's a larger question that I know all of our working groups and goal implementation teams deal with. But in that case, it can be helpful to be able to at least communicate the return on the investment that's been made thus far.
Do you think that it might be possible that there are benefits? Well, I mean, so yeah, there's certainly something to the scale I would envision that, like perhaps that more ecosystem benefits are gained through the sort of the mega projects, the big projects that are getting substantial investments, but that we still might be able to identify local benefits, for example, enhanced recruitment by some small tributary community, you know, supported projects and so on.
Yeah, definitely. And one thing that I think is important, where we are right now, I know a lot of work has been supported by the Bay program and by federal funding agencies to look at the ecosystem services of oyster restoration, and those projects have been super helpful in informing this. But I think the next step and the next level of that is to try and develop and understand some of the relationships about either the characteristics of the oyster habitat itself or the characteristics of the oyster population that are driving the responses that we see in fish utilization or productivity or whatever the ecosystem service is that we're interested in. Because I think one of the biggest challenges too is that these types of studies are also difficult to implement and expensive to implement. And so when you're thinking about smaller community-based studies, understanding things that could be measured as proxies for the ecosystem services that we care about, I think would be really beneficial, because it would give organizations and nonprofits like the one I work for, you know, the opportunity to quantify and talk about the return on investments of smaller projects if you don't have the expertise or the budgets or resources available to do some of these more advanced studies like the ones that Jeff is doing in quantifying denitrification and things like that.
Okay. So I'd like to shift the focus over to aquaculture and pose a question to Karen Hudson. I mean, clearly, I think we need to have a better idea of how aquaculture fits into the ecosystem. Do you have a sense that this is part of what's behind the societal concerns about aquaculture? If the communities on the shores had a better idea of how aquaculture fit in, would they be more inclined to support the activities, do you think? Is this limiting? Or do they just not?
That's a good question. Yeah, that's a good question. I guess my knee-jerk answer to that would be it's not going to matter. I think aquaculture, especially as people are as the industry is moving towards floating gear, things that are more obvious. I think if they, and aquaculture is near shore, right? So people can see it. And so I think it's separate. I guess my sense is that it's separate. I don't know that it will matter because it's so close. And we've lost, or we are losing and have lost the communities that understood what working on the water is all about, what waking up and seeing people, fishing on the water is all about. We've kind of transitioned into people coming from other places who want to own their view shed. And so I don't know, I guess my pessimistic view is it's not going to matter. But perhaps with some work and
issues might be overriding.
We could change it.
So a question while we're on aquaculture, Larry Sanford has a question. There's been a lot of discussion about restoration, but what about the ecosystem benefits of aquaculture? Are they the same? Are they different? Is there some mix? Clearly the financial benefits are higher for aquaculture. Does somebody want to handle this question? What do we know about the ecosystem benefits of aquaculture? Are they the same? Are they different from restoration, from what's happening in wild populations?
I can address a little bit of that. With on-bottom aquaculture, the parallels to restoration are a little bit closer, but the culture practice of moving oysters around and culling them all the time makes for a pretty different structure to that community. And so I think we recognize that there's going to be enhancement of what I look at, nitrogen removal, associated with aquaculture, both in the tissue, but also potentially in the denitrification with the on-bottom part of it. We have an N of 1 or 2 in that, and we don't really have a way to generalize to a much bigger picture. With water column aquaculture, it's a little bit more of a mixed bag in the sense that when you overload the sediments, there's not much of a water quality benefit, and there's some issues that are negative attached to that. But if you have a place with lots of dispersion of biodeposits, it could be extremely positive. So what we're faced with in terms of aquaculture is a lot of site-specific characteristics are going to determine, at least from a water quality perspective, where they fit into the spectrum from deleterious to highly beneficial.
Mm-hmm. So Jeff, so I think I just heard you say that water column aquaculture maybe would not be accruing as much of a benefit as the benthos. Do you think that's true as you expressed it, or is it just that it's harder to measure, and because of this dispersal of biodeposits and so on, we're not capturing it?
I think we're not capturing it. So the work that Larry Sanford and I and Jeremy Test and others did at one of our sites here suggests that 90% of the biodeposits move into adjacent areas where they're likely to be denitrified, and the water quality benefits are likely to be very, very high. But Bonnie Brown and others at Virginia Commonwealth, looked at a site where all this material focused in the bottom, and it took poor sediment quality and made it much worse by basically overloading the sediments with organic matter. And I focus on the water quality part of it, but there's also you can have negative impacts on the benthos, the benthic animals that are associated with aquaculture too. It's highly likely that the on bottom can have value, but in order to really credit the water column, measurements can help, but you really have to use a model probably to get at what the real value is.
So Mike mentioned the shell resources, which sort of reminds me of all of the discussion we've had of carbonate budgets and so on, which there's certainly evidence suggesting that we're struggling to maintain positive carbonate budgets in a lot of what we're doing in terms of restoration and managing the wild resources and so on. How do we deal with this? Does this suggest that we should be prioritizing other populations, shallow water systems, where there possibly wouldn't be the same level of taphonomic activity producing degradation of the shell resource and so on? as opposed to working in deeper waters where there's more burial and so on? How do we deal with the shell budget issues? Anyone have any thoughts on that, Mike or Allison?
Yeah, I've been thinking about this mainly in terms of thinking about what information I would want to have in order to try and model it. But it ends up being a really tricky question because there's nothing about oysters that's simple. If you think of the shape of an oyster, how they grow together, it all forms very complex shapes. And my at least current thinking on it is that though that level of specificity is likely important in how an oyster shell reacts with the environment. How much it's cemented together in big clumps limits the surface area for how quickly it can degrade. How much living materials on the outside that's protecting the inner core of a reef. And so trying to think about how generally oyster shell and oysters interact in the environment, as well as the rest of the community that they support, is pretty tricky. And I think that's why I think that the studies that have been done to date aren't necessarily sufficient to describe the dynamics that we have. So even things like how rapidly does oyster shell dissolve? If you just have oyster shell that you put down in the bay, I don't think we have a great idea of that. And it probably varies depending on where it is in the bay.
Okay. Juliette mentioned the complexities, the multiple stressor interactions. A number of you have talked about the challenges in addressing questions in a suitable way. Do we have the resources that we need? Do we have the expertise? Do we have infrastructure in place to address some of these questions? And I ask this because this, in my own area of parasitology and pathology, and this is something we're very much talking about. How do we take apart these complex systems, these complex interactions, and do we have the expertise and the infrastructure in place to address this when so much is moving, for example, to molecular genetics, where we're not doing experimentation anymore, we're just looking for eDNA and so on. Do we have the expertise? Do we have the facilities in place? Do we have the infrastructure to address these challenges? And if not, what do we need? What advice should we be giving the funding agencies, our institutions that are charged with keeping labs open or closed, and so on? Does anybody have any thoughts on that?
I'll just very quickly say that I don't think we take advantage of the synergistic opportunities that we have with all the various research and other programs that are ongoing to try and make these sorts of studies more cost effective. Mm-hmm. And so that would be the main thing I would look more for, is to try and use all the various restoration and management activities that are going on and figure out ways to use them in a more deliberate adaptive management strategy to try and improve our understanding more quickly.
That's a great point. Any other thoughts on that?
I'll just say that I think to Mike's point, there are a lot of opportunities out there for partnering with nonprofits and community organizations like ours, and probably have a lot of data sources that people are not aware of. So for example, we have an oyster gardening program where we've got over 1,000 people who are growing oysters at their docks throughout the entire Chesapeake Bay. And we keep detailed records on the survival and the growth of those oysters. And, I would encourage people to look at some of the organizations like Chesapeake Bay Foundation and others who participate in citizen science programs and utilize either the data that's coming out of those, or just recognize that there are organizations out there who have opportunities for people to connect you with field sites, or connect you with oyster growers who are interested in participating in research, or having a long-term data set on oyster growth from throughout the entire Chesapeake Bay. There are some opportunities out there where we may not know what research is going on and know where to plug it in. But I'll just put that out there as there are probably resources that you guys aren't aware of from citizen science and other, small scale research efforts as well.
So there's a comment from Bill Ball that just came through that connects to this aspect of the discussion. There has been discussion of the fact that many Chesapeake Bay projects have been at large scale and complexities associated with multiple interacting factors and stressors. In this regard, one might think that more could be learned from the large scale activities if there was more attention to better science during these, particularly giving thought to the conduct of parallel control experiments at similar scale. So I think that's, it sort of gets to the same point that maybe that Mike had and Allison sort of reinforcing that, thinking about the different activities that are going on, working to sort of integrate and find the synergies and put things together in a way that might create more power. And maybe to think maybe a priori about, how might we set these up so we can ask questions and get good answers coming out of these activities. I don't know if anyone else has any other thoughts on Bill's comment. Feel free to chime in if you do. And here's another question from Ray Najjar. It seems we cannot understand the fate of oyster shell biomass and the impacts of acidification on oysters and other shellfish if monitoring of the carbonate system by the Bay program is limited to pH. If we could add another variable like alkalinity, we'd be able to constrain the carbonate system and estimate, for example, the calcium carbonate saturation state. What do the panelists think about this? Any thoughts?
Well, I think, I mean, that could be targeted as well to areas where you're emphasizing the production of oysters on the bottom to see whether there's a limitation in those particular places. I think from Ray's talk yesterday that there hasn't been as much of this kind of measurement. It used to be a more routine measurement, and now it's not done as much, with the exception of some research projects like Wei Jun Kai at the University of Delaware. And it would be a rather useful thing, I think, especially in these larger scale restorations, to get an understanding of whether or not that the CO2 system is a stressor in terms of success.
Mm-hmm. Mm-hmm. Okay. And I would just sort of, along these same lines, echo earlier comments by Karen and Juliette that these, the issue of what might be stressing these animals, and I think particularly in early life history stages, out in the environment. I mean, we're building a profile of what's happening in hatcheries to some extent with regard to aquaculture production, but we don't have a good handle on what the aquacultured animals are experiencing once they're outplanted in the field. We don't have a good handle on larval and early life history stages of oysters and other marine bivalves with regard to any of these environmental challenges, the carbonate chemistry challenges, algal toxins, and so on. So that's certainly registering with us as a major knowledge gap.
If we go back to Bill Ball's question, so he was asking basically, could we, a nature of these kind of restoration projects is they're such large scale. When we work in Harris Creek, so much of the bottom was covered that, we have some control areas, but they're not maybe as effective. And so, sort of that idea, depending on the kind of restoration you do, it's a lot easier to commission barges and bulldozers than it is to think about a sort of a scientific design with controls, and think about the not just sort of the initial planting and immediate success afterwards, but to basically have a lessons learned approach to this. So as you move from large scale project to another, you have an idea of a lot more of the variables that control success. And I think there's the enthusiasm to get these things constructed and built, and you probably need to use the momentum to do that as they're being built. But I think, maybe we're not getting as much out of these in terms of understanding as that we should otherwise.
Thanks, Jeff. So I noticed we've got about 5 minutes left in the session. We've done a pretty good job with the questions that have come in. I would like to ask the panelists to take about 45 seconds or a minute each to make any final comments, share any final thoughts, perhaps with regard to the other questions that I've posed to you that we haven't had a chance to talk about. So let's start with Allison. Do you have any final thoughts as we move toward wrapping this up?
My only thing is I'm tremendously encouraged by all of the research and work that's been done. I think in the Chesapeake Bay region, we have a tremendous amount of expertise, and we are fortunate to be able to tap that. There's so many people working in oysters, in the oyster restoration space in this region, and I think that better communication, better coordination of those efforts can really help us amplify and fill some of these knowledge gaps, which are so critical to policy and management. And also just making sure that there's support for this work moving forward. As I mentioned before, from the perspective of policy, we need to be able to demonstrate that what we're doing is working and making a difference. And it's the goal and the work of everybody on this webinar who contributes to that. Thanks.
Thanks, Allison. Jeff, any final thoughts?
Yeah, I mean, thinking about the nitrogen crediting, and restoration, we're really tied mostly into how successful is a reef going to be. Is it self-sustaining? Does there have to be continuing investment? And I think, forgetting the area that I'm in, but it's that overall success of restoration that's really the driving force for this. What we're doing with water quality benefits is an addition on top of really the key benefits of trying to get these animals out there and to increase diversity and all these other things. And it's really, making sure that we're doing the things to understand whether or not we're succeeding in each of these individual 10 restoration sites is critical.
Thanks, Jeff. Karen?
I would just say, kind of echoing what Allison had mentioned, that, there's any benefit in any one of these areas, restoration, management, fisheries management, aquaculture, is gonna have co-benefits across, across all of these sectors. So I'm very encouraged of all the good work that's being done. And I think as we all work together and maybe better utilize, better network, maybe that we can make improvements in all of these sectors.
Thanks, Karen. Chris?
Well, thank you. So I think in closing comments, I'll just start with something Allison pointed to, was, working together, communicating. All of this is very beneficial. It sounds very basic, but as we work together and talk through these issues, it's better than combating and trying to run to the legislature, et cetera. So work together, communicate. I think too, also on the topic of research needs of this panel, the more we can refine our understanding of the restoration benefits, of course, the better we all are. We can communicate that message to the public. I think in some cases, there was some overselling of what all this would generate and do. So I think it's important to have some integrity, have some clarity, and just keep refining our understandings and communicating that to the general public. And then finally, really addressing shell. And whether we buy it, we make it, we help the oysters make it. Shell is being lost from the bay. Everybody knows that. We need to look at the other end of the equation. How do we get more into the bay? And I think addressing shell is going to be crucial for the future.
Thanks, Chris. Juliette?
I just have two small kind of comments, I guess. One is that I've heard mentioned, and I've learned a lot during this panel myself, about kind of the larger projects that are occurring within the bay about restoration or understanding the system. And so I've also then heard kind of tacked onto that the idea that, you know, it'd be great if we could add more monitoring of more parameters on that, those efforts that are already occurring, so that we could kind of monitor that as it's happening, and I am all for that. I guess the only thing that I would point out is that from a research standpoint, that's moving towards like 5-year, multi-million-dollar projects, which I'm also a fan of. But the idea would be that we kind of have to move, I think, the funding agencies to kind of understand that. So whoever, you know, does maybe have an ear of program managers to be thinking like how, by moving to these 5-year, multi-million-dollar projects, we could do these very collaborative, you know, multidisciplinary, multiple expertise coming together to answer these really complicated questions. And my only other plug is that within hatcheries, you know, it was said that currently we don't really feel like we know the one thing that is impacting hatchery production. I agree with that, except that I would add to that, we most likely don't know the multiple stressors that are coming together to contribute to hatchery production reductions. And that I hope everyone continues to move forward with multi-stressor experimentation and monitoring, looking towards those very sensitive life stages that are the hatchery itself. And we continue to build those great relationships between industry and academia, because without those, that research can't move forward. All done.
Thanks, Juliette. Mike, 15 seconds for a final thought.
Sure. So I'll just echo on Allison's comments that I'm really optimistic in the progress that's been made recently, and that we can continue that momentum forward. And the last thing I'll add that I'm still surprised didn't come up, but I think understanding how diseases will continue to affect oysters in the future is a major thing that we need to continue to understand. And I think that low disease mortality has been one of the reasons we've seen as many positive increases as we have in recent years.
Yeah. Okay, thanks, Mike. And thanks so much to all the panelists for their contributions during this hour. This was great. And thanks to the audience for plugging in. And with that, we'll close the panel. Thanks to all of you.